Thank you everyone for joining the webinar today. So this is the second part of our three-part webinar series that we've been running over the last month or so. So my colleague Rain did the first talk, which was from Sample to Success, NGS Foundations with Agilent Omega and Element. Today I'll be doing Exploring NGS Workflows Across Vendors, and my colleague Ross in a couple of weeks will be doing an introduction to NGS data analysis. So before we begin, I just want to show you where diagnostic fits in. So we supply for a range of companies in the molecular space, all the way from sample prep right through to sequencing and data interpretation. As you'll see on the slide, we distribute for a number of genomics companies such as Agilent and Twist, which I'll be delving into more detail later. We have also recently started distributing for Omega Biotech for all your extraction needs. And we have Element now to do sequencing. And also we have partnership with Seq1 and Hyrex to do data analysis. Today we'll be exploring library prep solutions from several vendors, particularly how they contrast with one another and the strengths and weaknesses of each and where they would fit into your particular workflow and experiment. So we'll be looking at Agilent Technologies, Watchmaker Genomics, and Twist Biosciences. Now, before we begin delving into the different workflows, let's just look at an overall summary of an NGS workflow. And this is without enrichment. So what is a library? So a library is a collection of DNA fragments prepared for sequencing by performing a series of enzymatic steps. These steps are commonly referred to as the library preparation. So you'll have your isolated gDNA or RNA. Obviously, if it's RNA, you need to convert it to cDNA before proceeding in the library preparation. You then need to share it or fragment it. Normally, this is done either mechanically with a bioreactor or a Gavaris, or you can do it enzymatically as well. For example, Agilent uses enzymatic fragmentation. After fragmentation, you'll have ends which are not even with each other on the double-stranded fragments. You then need to repair these ends. So you need to generate blunt end fragments. So you just fix it there or repair it. And then you need to do A-tailing. So this is adding an A base to the 3' end of each strand. And this is so that you can add your sequencing adapters. So what are your adapters? These are short DNA oligos that contain the primer sites used by the sequencer to generate the sequencing read. Okay, so this is, for example, the P5, P7 used by Illumina sequencers. Adapters can also contain short 6 to 8 base pair sequencing sequences called indexes or barcodes. So these barcodes allow you to multiplex different samples in the same run. Okay, so it's a unique ID for each particular sample. You add your adapters through various PCR reactions, and then you use a PCR to then amplify your library so that you have more of it to proceed in the sequencing. And then at the end, you've got your whole genome sequencing library. Now, this is before doing any sort of target enrichment where you would select for specific fragments within this larger library. So just an overview of NGS library prep. You have, I guess you could say, 3 different ways of doing it. You could have it with no target enrichment. So essentially you extract your sample, so DNA on RNA. Remember, you need to convert the RNA to cDNA. You do your library preparation and then you proceed to sequencing. So this would be whole genome sequencing. You could use this for whole transcriptome analysis, or RNA-seq, or shotgun metagenomics. You could do it with target enrichment, so a different way of preparing the library. So essentially you extract your sample, you do your library prep, but then you capture using specific probes that are targeted for your genes of interest. You pull those out of solution, and then you proceed to sequencing on that smaller subset of the greater library. So this is useful for pathogen capture from mixed samples, whole exome sequencing or custom gene panels, and targeted RNA-seq. And then lastly, you could do target enrichment, but through using amplicons. So once again, sample extraction, PCR amplification in this case, Then you proceed to library preparation on those amplicons, and then you would do sequencing. So this is used for 16S RNA sequencing, T cell receptor or B cell receptor sequencing, or viral sequencing. So it all depends on your costs and what you are trying to answer. So the choice between amplicon-based and hybridization capture-based NGS target enrichment depends on specific research goals, what your budget is, and the sample types you have. So amplicon-based methods are normally ideal for smaller panels and known variants, offering simplicity and cost effectiveness. In contrast, hybridization capture or target enrichment is better suited for comprehensive studies requiring broader coverage and the ability to detect rare variants. Now we'll jump into library preparation for the first company we'll be discussing today, and this is Agilent. So just a little bit of background on Agilent. So this is a US company headquartered in Santa Clara, California. They're a spin-off company from Hewlett-Packard, yes, the printing company, and they were actually established in 1999. So they're not that old, but they've been around for a few decades now. They provide instruments, software, services, and consumables for laboratories in the life sciences, diagnostics, and applied chemistry markets. People in the audience may be familiar with their QC instruments, such as the bioanalyzer, tape station, and fragment analyzer, or you may be familiar with their liquid chromatography portfolio, which we do not distribute that particular portfolio, but you may be familiar with those instruments. So essentially, they provide NGS library preparation solutions using their SureSelect chemistry, and these are target enrichment specific workflows. So I'll be going into detail on two library prep solutions from them. This is their XTHS2 chemistry and their MAX chemistry. So I'll start with XTHS2. So this is a newer Agilent library prep chemistry released after 2020. It's a refinement of an older XTH chemistry. So there was an XTHS1. And essentially, it's a target enrichment-based library prep. It allows for input amounts of DNA ranging from 10 nanograms to 200 nanograms. Various quality of DNA sources can be supported from blood all the way to FFPE. It supports mechanical and enzymatic shearing. There's also pre or post hybrid capture pooling protocols available, which I'll describe a little bit later what that is. It allows for 384 index pairs for dual indexing. And there's also double-stranded UMIs available as well for PCR and sequencing errors or error correction. So this is just an overview of a typical XTHS-based workflow or the SureSelect workflow. And essentially, let me get my pointer. Essentially, we have three sections to it. So you've got the design process. So if you are doing a custom panel, so one that you are designing, there's a specific software, online software that you're able to use in order to design your panels of interest. Or if you are using an already made panel, you can then order that, and then you can go into the library preparation, and this entails extracting your DNA, fragmenting, barcoding the DNA, and then you have your prepared whole genome sequencing libraries. You then proceed to target enrichment, which is the second step, or your post-capture steps. And this could include this includes hybridizing your panel, essentially your probes to your genes of interest. And this can be done as a fast method, so less than 90 minutes for hybridization, or it can be done overnight. So there's two different flavors to the protocol, depending on what you are doing and which protocol you are using. You then have your captured libraries. You then need to amplify those, and then you would proceed to sequencing. Also in this workflow, there's also different workflows depending on if you are pooling your libraries before you capture your sequences of interest or afterwards. And I'll explain after this what this entails. So a little bit on target enrichment. What is it exactly? What is happening with the Agilent chemistry? And essentially, you are using RNA probes to capture genes of interest or gene targets of interest. Essentially, these probes are cRNA probes. They're 120 nucleotides long, and they are bioatenylated. Now, this is important because this biotin on the probes allows you to use stripped avidin coated magnetic beads to pull your probes, which are bound to your targets of interest, out. And then you're able to enrich or isolate your genes of interest. Okay. So this can either be done overnight, and also there's a possibility to do it using a 90-minute fast hybridization protocol. So there's both for XTHS2 and also for the max chemistry, which I'll describe a bit later. So what are they doing for their probe selection? How are they designing these probes? So they are selected on known manufacturing capability or characteristics of the probes. They also optimized for target capture, and they efficiently observe improved performance. They reduce the design footprint. They improve uniformity, and there's also fewer off-target reads when they're designing these particular RNA probes. So why would you do target enrichment? What is the pro of doing target enrichment or hybrid capture? So essentially, you're only sequencing your desired regions of interest. So this could be exons, this could be particular gene panels, or this could be intergenic regions in the genome. Essentially, you would then open up more capability for multiplexing. So you will be able to sequence more samples per run compared to whole genome sequencing. So, for example, if you are only doing exomes, that's only 1% of the entire genome that you're now sequencing. So obviously, you've now opened up more capacity on your sequencer for more samples. It's also smaller data sets. So you would need less intensive computational analysis, and there's probably a faster turnaround time as well. And you have increased confidence and accuracy in variant calls due to higher depth of coverage in regions of interest. And also, it's more focused. So you would have less variants of unknown significance. So it's more targeted. Most of the currently known drivers of mutation or driver mutations, so cancer-causing mutations, affect the coding sequences of proteins or protein coding genes. However, protein coding exons only account for 1% of the human genome. So performing whole genome sequencing is not always cost and time effective. So whole exome sequencing is attractive for clinical applications, mainly because it covers actionable areas of the genome and can identify disease-causing mutations in protein coding genes. So this is just explaining post and pre-capture pooling, what is it exactly, and what are the strengths and weaknesses of either one. So with pre-capture pooling, essentially you have your 8 or 16 library preps as an example. So you've got 8 or 16 separate tubes. You would then pull all of those into a single tube, and then you would proceed to your capture. In post-capture pooling, you've got your individual library preparations, and you maintain them in single individual tubes. So you would do your captures individually or independently. And then afterwards, you would then pull the samples. So now there's pros and cons to doing it either way. So the benefits of pre-capture pooling is that you would have reduced reagent costs, and there's less tubes to process. So it's a bit easier to do it this way. The downside is that you could have variable uniformity of reads across all samples, because now you're combining all these different samples into a single tube, and you're trying to do the capture all at once. So obviously there's some effects on the stoichiometry within the tubes, then the single tube. Yeah. And then this is also just illustrating what a final show select library would look like. So this would be for XTHS2 chemistry and also for the max chemistry, which I'll explain in a couple of slides. And essentially, you could prepare it or two flavors of the libraries. You can either have molecular barcoded tag libraries or molecular barcode free libraries. And essentially, the final library would have your insert here in blue. You'd have your UMIs or molecular barcodes on either side. You'd then have your indexes in in red, and then you would have your adapter sequences in yellow, which would bind to the flow cell surface. Also want to illustrate with XTHS2, there's also a RNA workflow for XTHS2 and for MAX as well. And essentially, you can do mRNA enrichment with these workflows or total RNA. Currently, there is no RNA depletion capability with these workflows. And also want to illustrate that before the library preparation, you'd have two tracks. So if you are doing DNA and RNA in the same workflow, for DNA, you would have your mechanical enzymatic shearing, and then you'd proceed to library prep. With RNA, you would fragment, and then you'd have your cDNA synthesis. And then essentially you would have identical steps for the library preparation afterwards. So it makes it much easier to process these samples, as the workflow would then mirror each other. It would be much easier to then process all the samples at once after these particular steps. Now we move on to the SureSelect Max chemistry. So this is the newest library prep chemistry from Agilent. Essentially what they've changed here from XTHS2 is that they are using modular library kits now. So this allows more flexibility for the end user. So you've got standalone beads, adapters, primers, and blockers, for example. The protocols have also been streamlined, resulting in reduced library prep times. They've also improved the performance over previous Agilent chemistries. So there's improved coverage depth and uniformity, post-capture yields, and decreased PCR duplicates. It also now officially supports low-cycle PCR whole genome sequencing workflows, and there's now official MGI support, so DNA nanoball sequencing without the need for any sort of conversion kit. So this just illustrates the turnaround times for the particular formats of the different SureSelect Max kits. So you've got for DNA using enzymatic fragmentation, you've got DNA manual shearing, you've got total RNA, and then mRNA. You can see the turnaround times is 6.5 to 8 hours for both the enzymatic and manual shearing protocols. For RNA, it's 9 to 10.5 hours. And then for mRNA, it's 5.5 hours. So what they've done here in order to improve these turnaround times is that they're using one-step fragmentation, end repair, and detailing steps. They're now using master mixed reagents. There are faster ligation times now in the protocols, faster PCR cycling conditions as compared to XTHS chemistries. And they've also reduced the bead capture times as well in order to see these time savings. So this is just a table that I've put together comparing SureSelect XTHS2 and SureSelect Max. And I just wanted to show you what the main differences are between the two library chemistries. So you can see for the turnaround times, if you are preparing either the DNA for Illumina libraries, MGI, RNA, mRNA, or whole genome sequencing, you can see there's time savings for the Illumina compatible libraries. There's also time savings for total RNA. And you can see they've now introduced a DNA preparation for MGI and officially support whole genome sequencing as well. Also, another key benefit is that there's improved overall capture performance versus the older XTHS chemistry. And also they've optimized, if you are using the pre-cap pooling protocols, it's also been optimized versus the older chemistries as well. I just want to mention SureDesign as well. So this is an online free platform for creating custom SureSelect target enrichment probe designs based on your gene targets of interest. So you would define the regions of interest and you would select the probes based on your target genes, your genomic coordinates, or your selection parameters. And then you would be able to use the platform to design your own custom panel. Essentially, SureDesign provides you with flexible, adaptable software for probe design on all Agilent genomic platforms and applications. It's a guided, easy-to-use precision workflow for creating your custom designs, leading to NGS probe selection algorithm for improved sequencing data, and you have access to empirically validated catalog probe designs from the SureSelect libraries that you can use as well. And essentially, it's quite easy to use. You would log into SureDesign. You would choose your technology. So in our case, we are discussing NGS, but it's also available for CGH and CRISPR. You would identify your genomic regions of interest. You would input your chromosomal locations or your gene IDs. You would choose your default optimized parameters within the software, or you could input custom parameters within the software. You would review and submit the completed design. You order the product, and then you would receive your custom design. I also want to highlight that for the custom designs, Agilent uses a tiering system. So depending on the size of the tier, it would change the price to you as the customer. So they've got essentially 6 tiers. So it's a tier 1 to tier 5, and then a large tier which is a panel design which is greater than 24 megabases. So also want to highlight the two NGS panel designs that are using XTHS2 or max chemistry. And essentially these are, you could say, the flagship panels from Agilent. This is their Sure Select Human All Exome V8 panel. So this is an exon panel targeting 35.1 megabases of content, and it targets 99.7% coding exons, or of coding exons. And then the other panel that I want to describe briefly is the Sure Select Cancer Comprehensive Genomic Profiling, or CGP. And essentially, with this panel, you are able to sequence 679 genes, and you're able to detect single nucleotide variants, copy number variants, insertions, deletions, translocations. You can also calculate TMB and also analyze MSI. And there's also an RNA component where you're targeting 80 genes, and you can detect de novo gene fusions with this particular panel. So both panels are compatible with libraries prepared with Sure Select, XTHS2, and Max chemistries. So the first one I'll go into detail with is the CGP panel. So essentially, Agilent is offering the most comprehensive commercial pan-cancer NGS panel with the CGP panel from them. And this is essentially for solid tumor profiling. It's compatible with both XTHS2 and Max library prep chemistries. It's got parallel DNA and RNA workflows, as I mentioned previously. And it's also available in automation or in automation solutions as well. So there's different cancers that you can target. So lung, breast, prostate, colorectal, gastric, bladder, kidney, melanoma, and pancreatic. And then the summary table on the right-hand side, I just want to highlight again the number of genes that you're targeting. Remember, it's 679 genes, so it's currently the most comprehensive pan-cancer panel on the market at the moment. You're also targeting 80 genes, so RNA in the RNA workflow component. And then the variants detected, once again, SNVs, indels, CNVs, etc. If you are interested in seeing what the gene targets are, there is a link down here below. I can always share the slides at a later date with you if you're interested, and you can go to this link and you'll see all the gene targets that are available in this particular panel. I just want to give you an overview of an end-to-end solution that would use the CGP panel. And it's also something that we set up in one of the largest diagnostic laboratories in Johannesburg, and they're currently using this end-to-end solution to great success. And essentially what this details is a workflow. So you could either use fresh, frozen, or fixed tissues as starting material. This is using XTHS2 chemistry, but it's automated, or the library prep is automated using one of Agilent's automated library prep instruments. So it's called the Magnus. Sequencing is then done on the Element VT platform, which is an instrument that we are currently distributing. And data analysis is then handled by Sequan, which is a bioinformatics company which is based in France. If you're interested in seeing this workflow and learning more about it, you can read about it in this white paper that I mentioned here at the bottom. Then the other panel that I want to overview is the exome panel from Agilent. So Agilent is offering one of the most widely used and trusted human exome panels on the market. And essentially here, their core panel, or the exome V8 panel, this is you could say, their bread and butter panel. And this is looking at protein coding regions from RefSeq, CCDS, and gene code, and also TRT promoter regions. You can see it's 35.1 megabases that it's targeting, but the entire panel is 41.6. And then you have other, you could say, flavors of this particular panel. You have one, for example, the non-coding clinical variants panel. This is very useful in clinical research. You can see it's looking at clinical variants, including pathogenic and likely pathogenic variants. You also have other panels which include untranslated regions, CNV backbones, mitochondrial genome, or pan-cancer biomarkers as well. Okay, the next supplier that we'd like to delve into is Watchmaker Genomics. So a little bit of background on them. So Watchmaker Genomics is a biotechnology company headquartered in Boulder, Colorado. And they actually have an R&D facility in Cape Town, South Africa. So they design, develop, and produce solutions for molecular biology, the molecular biology industry, including advanced enzyme engineering and RNA DNA library prep kits. So they offer various NGS kit solutions. So I will highlight two of them essentially, or three. So it's DNA library prep kits that they offer, and they also specialize in RNA NGS solutions. Two in particular, the mRNA and the RNA library prep with Polaris depletion might be of interest to you. So the first one I will detail is the DNA library prep kit from them. So it's designed for NGS library construction using Illumina sequencing technology. So essentially it's adding P5 and P7 adapters. It supports single-ended and paired-end reads. You are generating libraries normally with fragment sizes of between 200 and 600 base pairs with this chemistry. And these libraries are cross-platform compatible. Okay, so they also work with MGI, Element, Singular G4, and Ultima Genomics. Essentially, with this particular chemistry, it's low bias, it's high sensitive for accurate variant detection. It's also a very fast workflow, so you're able to pair libraries using as little as 500 picograms of input. So you can already see contrasting this with Agilent. This kit might be more useful if you have very low amounts of starting material. It's also an enhanced coverage of low frequency variants in cell-free DNA and liquid biopsy samples. So it's a very good kit for that. And enzymatic fragmentation enables consistent results from FFPE and low input DNA. And it's also automation ready and available in customizable formats. So this image here at the bottom is just showing the turnaround time for the DNA library prep kits. So you have one with fragmentation and then one without fragmentation. And you can see you're able to complete these library preps in under 3 hours for each of them. So this is just a table contrasting the library prep kits with fragmentation and then without fragmentation. I won't delve into too much detail here in this table, but you can see it later on when we share the slides. But essentially, I want to highlight adapter compatibility. It's compatible with full-length adapters or stubby adapters or adapters with or without UMIs. I also want to highlight here what sort of applications you would use these kits for. What are they compatible with? So what sample types? So for the prep kit with fragmentation, it's compatible with FFPE, genomic DNA, plasmids and vectors and long amplicons. And for the library prep, the normal library prep kit, it's also compatible in addition with cell-free DNA, chromatin immunoprecipitation DNA, bisulfite converted DNA, and short PCR products as well. Then this is just highlighting the application areas for these kits. You can see there's a range of different applications from whole genome sequencing to targeted sequencing, somatic and germline variant detection, copy number variation analysis, metagenomic analysis, bulk RNA-seq, viral sequencing, ChIP-seq, and fragmentomics. Okay, now we're moving on to the RNA library prep kits. So these are essentially, you could say what Watchmaker really specializes in and what they're really good at, I would say. And they have 3 RNA library prep offerings. Essentially, we'll only discuss 2 of them. And essentially, this is the mRNA library prep kit and then the RNA library prep kit with Polaris depletion. So this slide is just to highlight what exactly we are looking for when we're looking at total RNA and what are we trying to sequence and what question are we trying to answer. So in a typical RNA sample, the majority of RNA, so normally around 80 to 90% is ribosomal RNA, which forms the structural and functional core of ribosomes. messenger RNA represents only about 1 to 5% of the total RNA extract, but it's the key focus of most transcriptomic studies since it's what codes for proteins. The remaining fraction consists of various non-coding RNAs, including non-coding, transfer, intronic, and intergenic RNAs, which play regulatory or structural roles. So for sequencing applications, rRNA is usually depleted in order to enrich for mRNA or other informative RNA species, or you may enrich for mRNA directly as well, depending on which workflow you are using. It's also important to note that globin mRNA within the mRNA fraction could also make up about 70% of the mRNA fraction, normally when you're extracting from whole blood. So you just need to keep that in mind as well if you are using whole blood. So this diagram just illustrates what happens when you are enriching for mRNA or when you are depleting rRNA, and what applications would be more valuable for either one. So you've got bases on the y-axis, or percentage of bases, and then on the x-axis you've got total RNA. So if you have no enrichment done or no depletion done, then you've got if you've done mRNA capture, or if you've now depleted for rRNA. So you can see in a total RNA sample, most of it is rRNA, so in the purple 95%. If you then do mRNA capture, we are pulling your mRNA out of your total RNA sample. You can see then that most of it, assuming if it was whole blood, it would be mostly globin and exonic mRNA, so 59% and 29%, and a small fraction could still exist of rRNA, about 6%. Then if you do this Polaris depletion, which is part of Watchmaker's kit, you're essentially removing rRNA, and you can see now the full fraction is now mostly consisting of all your RNA except your ribosomal rRNA. So the rRNA library prep kit with Polaris depletion. So this enables preparation of stranded whole transcriptomic sequencing libraries. You can prepare libraries from as little as 1 nanogram up to 1 microgram of starting material. You deplete cytoplasmic, mitochondrial rRNAs, ETS and ITS human rRNAs. So ETS and ITS, these are non-coding spacer regions found in the precursor ribosomal rRNA transcripts. And you also deplete globin RNAs. The workflow is compatible with human, mouse, and rat species, and it's also compatible with high and low quality samples, including FFPE. So this diagram just illustrates how the Polaris depletion actually happens. How is it depleting the rRNA? So on the left-hand side, you've got your total RNA fraction. So you've got rRNA in globin, mRNA in blue, and you've got your transcripts of interest in green. You then add single-stranded DNA probes, which are specific for your rRNA and your globin mRNA. So this is an orange, and you can see it's binding to the blue. You then introduce this RNase H, and essentially this degrades hybridized RNA to DNA. So anything that is bound to the probes, the DNA probes, will then get degraded by this RNase H. If you degrade it, you've now enriched for your mRNA and long coding RNA, which is left behind, and then you would then degrade your single-stranded DNA probes. So what sort of application areas would this be useful for? So you could do gene expression analysis, you could do isoform, splice variant, or gene fusion identification. You can do single nucleotide variant detection. You can do novel transcript discovery, or you could do targeted sequencing protocols employing hybridization capture afterwards, or whole transcriptome sequencing as well, obviously. And then the other RNA library prep kit that I want to highlight is the mRNA enrichment library prep kit. So it's designed to enrich for mRNA. So using the poly A tail, which is unique to mRNA. And this is for eukaryotic mRNA and some non-coding RNAs, which have this poly A tail. It's optimized to produce target library insert sizes averaging between 200 and 300 base pairs. Its total RNA from eukaryotic species would be your starting material, so not bacterial or archaeal, as these lack poly-A tails. You would start with highly purified total RNA, so 2.5 nanograms all the way to 1,000 nanograms. It also requires highly intact or good quality RNA. So if you were doing a tape station or bioanalyzer, you would be familiar with the RIN number. But essentially, a RIN number greater than 7 means that it's a very high quality RNA. And this is essentially used for downstream mRNA enrichment. The workflow is essentially the same as the normal RNA library preparation kit with Polaris depletion. So what exactly is going on in this kit? What are we enriching for? How is it working? So if we look at the structure of a typical mRNA fragment, you've got your 5' end and your 3' end, and your poly-A tail is obviously on your 3' end, and it's normally about up to 250 nucleotides in length in mammalian RNA. And essentially the kit is using beads to capture these mRNA fragments. So you're enriching for protein coding RNA by hybridizing a poly-T primer, either in solution or immobilized on a bead. So essentially you have a poly-T primer on a bead, it attaches to the poly-A tail on the mRNA, and then you are able to enrich for your mRNA. And then afterwards, you're able to proceed to cDNA synthesis. So essentially, you would fragment or prime, you would have your first strand synthesis, you'd have your second strand synthesis, and then you would have your double-stranded cDNA library at the end to proceed to library preparation. What sort of application areas would you use this mRNA library prep kit? So you would use it for gene expression analysis, gene fusion detection, isoformal alternative splicing analysis, novel transcript discovery, single nucleotide variant detection, or some other pathway analysis. This is just a nice summary table comparing the two RNA library prep kits that I've just described. In gray, there's also just their normal total RNA library prep kit, which I haven't gone into too much detail there in gray. But essentially, I just want to highlight the RNA input and the library prep times. So there's a wide range of RNA inputs that you're able to use. So in this case, 1 to 1,000 nanograms for Polaris depletion, and for mRNA, 2.5 to 1,000 nanograms. And you can see the library prep times are quite short. So 4.5 to 5 hours, depending on which kit you are using. Okay, now we're on to the final supplier that I want to discuss, and this is Twist Bioscience. Some background on them. So this is a biotechnology company based in San Francisco, California. They were founded in 2013, and they specialize in manufacturing synthetic DNA products with specialized oligo printing technology. So this is their specialty. They essentially have NGS library kits that are developed with target enrichment in mind, and also with high throughput applications in mind, which is what I'm going to discuss in a couple of slides. So they have a wide offering of solutions in the NGS space. So they offer a wide range of options. They have fixed panels. They also have custom panels that you can design with them. They have various library preparation solutions. They have hybridization and indexing solutions for labs. They also have methylation detection systems, and they also offer synthetic controls. This is just showing what sort of NGS target enrichment panels they are able to offer. This is not the full gamut of everything that they offer, but essentially they have their own exome panel. They've got a very interesting mitochondrial panel. They have two viral panels that I want to highlight. There's a respiratory specific panel, and then a comprehensive viral research panel. And they also offer solutions for long read sequencing target enrichment. They also have methylation detection systems, or a human methylome panel. And then for transcriptomics, they also offer their own RNA exome preparation panel. and then just general RNA library preps as well. There's obviously many more panels from them, and this is just sort of a small snapshot of what they can offer in terms of target enrichment or NGS panel solutions. So I want to highlight their high throughput solutions. I think this would be of interest to you. And essentially, there's two kits that I want to highlight. So the first one that I want to highlight is the Flex Prep ultra high throughput NGS library preparation from them. And essentially, this kit offers an NGS alternative to traditional microarray based technologies for population studies, agri-genomics, and other ultra high throughput applications. So they use a technology called normalization by ligation technology with enzymatic fragmentation methods. The kit offers built-in sample normalization across a wide range of DNA inputs, reducing both cost and complexity of typical sample processing steps. So early sample barcoding enables downstream pooling of 12 separate samples into one reaction for more streamlined and efficient workflow. So I'll highlight this in the next few slides, how this kit exactly works. And this is just the specifications of this kit. So primary applications would be for whole genome sequencing or low-pass whole genome sequencing and genotyping. The sample input type would be genomic DNA, and the library prep is quite quick at 5 hours. But you can see the DNA input is quite larger compared to the other vendors that we've highlighted today. So you would require at least 30 to 300 nanograms of starting material. The fragmentation method that they use is enzymatic. Indexing method is unique dual indexes. And you're able to do target enrichment on up to 96 samples per target enrichment reaction. So you can see already that that's high throughput. You can enrich 96 samples at a time. And the total samples per kit, it depends on which kit you purchase, but it's either 192 or 1,152 samples, depending on which kit configuration you order. So how does this kit work exactly? So in a typical enzymatic fragmentation library prep for 96 samples, so you've got a whole plate with 96 samples, you would quantify and dilute, you would fragment your samples, you would do your adapter ligation, you would do spree cleanup, a PCR, another spree cleanup, and then you've got your 96 libraries that you need to QC and then dilute at the end. With the Flex Prep ultra high throughput library prep for 96 samples, essentially you are using this normalization by ligation technology, which eliminates the upfront quantification and the normalization. And essentially how this is working, and it's a proprietary system, so there's not too much in-depth detail on this, but essentially it combines enzymatic fragmentation, adapter ligation with inline barcoding, and early sample pooling to create a more streamlined, high-throughput workflow that reduces costs and reagent use. So you can see here you fragment, and then at the ligation step, you barcode the samples during the ligation reaction, and then you pull prior to the first cleanup. So you can see you're going from 96 to 8, so 96 individual wells to 8 samples. And then this is highlighted in this slide, where it shows that each row of a 96 well plate is then pulled into one tube. So you start with a 96 well plate, you've got 12 inline barcodes essentially, and then this is pulled into a single tube. So you'd have 8 tubes of 12 samples in each. Once these libraries are prepared, you can then move on to whole genome sequencing, or you could proceed to target enrichment. So very, very useful and good for high throughput sequencing. I also want to highlight the cost savings and the plasticware savings. Essentially, you would have less ethanol that you would use, less pure bead mix for post-ligation, as free beads and ethanol as well, much less. And you can see much less tips that you are using. So for standard kit, they highlight 17,000 tips that you could possibly use for the throughput of samples that you are doing. So this is based on an 8 well versus 96 well workflow versus 3,936. So quite a cost saving, plasticware saving, and a reagent saving. as well to you, the consumer. So what are some of the application areas for this ultra high throughput library preparation? So this could be high throughput genotyping, so designed for large scale projects, enabling high confidence variant calling from reduced sequencing coverage. It could also be used for human population genomics, so you could efficiently support human genotyping studies across large populations, offering an advantage over traditional arrays. And it can be very useful in agrigenomics. So it's cost-effective genotyping and market discovery. So in agricultural species sequencing, so for example, in bovine samples. And it could also have applications in precision breeding, genomic selection, and parentage testing as well. Now, the other kit that I want to highlight is called the 96-plex library preparation kit. And essentially, this is another high throughput kit that they are offering. And with this kit is that there's no fragmentation, end repair, or ligation involved in this kit. It's tunable for high or low GC content samples, and there's little to no automation needed for this particular kit. And you are able to do either 96 or 960 individually barcoded samples. So the Twist 96-plex library preparation workflow starts with up to 96 individual samples in a plate. After the first random priming and barcoding steps, all the samples are pulled into a single tube for the rest of the process. An optional plate barcode can be added in the index read position to allow simultaneous sequencing of multiple sample plates. So this slide here is highlighting you start with a 96-well plate. Each sample is individually barcoded. You then pull into a single tube. And depending on the kit, you could pull up to 96, so 10 96-well plates. So if you have 10 plates, this would be 10 tubes, which equates to 960 samples. So they currently have two kit configurations. It's either 480 samples, 5 plates, or 960 10 plates. So some of the application areas for the 96-plex library prep kit, these would be genome-wide association studies, diversity analysis or population stratification studies, SNP studies, fine mapping or SNP discovery. You could be detecting deletions, insertions, or rearrangements. This works with or without a reference as well, so application areas there are useful. So it's useful in agriculture and the pharmaceutical industries, such as seed selection, cattle breeding, drug design, or plasmid sequence verification. And then I want to highlight, Twist also has a partnership with Element. So Element is a sequencing company, and they offer a benchtop sequencing solution called the AVT, which is something that we distribute. And there's currently 3 AVTs in South Africa at the moment. And essentially, what they're offering here is something called Trinity. And essentially, this is target enrichment, but on board of the sequencer, which is quite advantageous. So if we look at the top of the slide here, normally with a target enrichment reaction, you've got your DNA library, and then you have your target probes. In a typical library preparation, if you're using an overnight hybridization, it normally takes about 16 hours, and then you have to do a certain number of washes afterwards, which can be quite laborious. And if the user is not experienced, there's a lot of room for error during these washing steps and this hybridization. This traditional workflow can be multi-day with several laborious steps. Now, essentially with this Trinity chemistry, what's happening is that you are doing the hybridization off board. It's a fast hybridization, but then you do the capture and the washes and the capture on the system itself. So Trinity enables less than a one-day workflow, cutting down hands-on time in half and accelerating time to results. Now, there are some panels that have been released. So at launch, it was an exome-specific panel with a spike-in of additional probes. And there's also a human 600K panel, which is a broad SNP panel for human population genetics research. So With this Trinity chemistry, you can detect minor allele frequencies down to 0.5%. You also unlock a greater processing capacity. It's also superior performance for insertion deletion accuracy of 0.99. And there's also a partnership with IDT as well. So it's not only Twist as well offering these kits. You are able to, there's also a partnership with IDT as well. So we get to the end of the presentation. So in summary, Agilent offers comprehensive and customizable enrichment solutions for NGS, including first-in-class CGP cancer and exon V8 panels. Watchmaker Genomics offers specialized workflows for challenging RNA samples, including their Polaris depletion kits to efficiently remove abundant, non-informative RNAs to improve the detection of mRNA and non-coding RNAs of interest. Twist Biosciences has efficient offerings for ultra-high throughput NGS sampling, reducing manual steps and maximizing throughput with agricultural and pharmaceutical applications in mind. And then just some things for you to think about also. So end users, so you as the audience member, have multiple high-quality NGS solutions now available to you to suit your budget and your throughput needs. You should also clearly understand your research question. So what sample type are you working with? Is it DNA? Is it RNA? Are you working with very poor or degraded samples? And what is the required scale before evaluating any of the solutions? And don't be afraid to ask us, the vendors, for help. So always leverage vendor support to clarify protocols and confirm the best kit for your specific application needs. So that we come to the end of the webinar. So now we've done 2 of the 3 episodes that we have planned. And I'd just like to highlight that my colleague Ross will be doing a presentation in a couple of weeks about NGS data analysis. So that'll be on the 25th. And thanks very much. I'll take any of your questions. I'll also check in the chat if there's any questions. If you don't have any questions now, you can also drop me an email, and I'm very happy to answer any of your questions as well. Thanks very much.